We're going to finish this lecture speaking about mutation. Let's start with new terms. The first one is genome. The genome, it is an entire information, genetic information, which is written down into chromosome. It is an essential information of the cell, and you remember probably already that bacterial chromosomes are circular versus linear or X chromosomes in humans. A lot of bacteria have additional small circular chromosomes located in cytoplasm. What kind of information will be stored in the plasmids? In the plasmids, usually bacteria store non-essential information. For example, at certain period of life, at certain point of life, bacteria become resistant to certain antibiotic. This information considered to be not essential for the cell. That means it will be written down into plasmids. The genome of any organism can be considered from two different points of view: genotype and phenotype. Genotype describes the cell's genetic plan. It's actually what it is written down into genes. Phenotype describes the effect of the plan on the cell's appearance and function. Mutations. A mutation is any chemical change in a cell's DNA. You have to know 5 different types of mutations. Those are deletion, inversion, transposition, duplication, and base substitution. Deletion means one pair or few pairs of nucleotides are deleted from the DNA molecule. Inversion reverses order of nucleotides within DNA molecule. Transposition takes one or few nucleotides from the region of DNA molecule where they're supposed to be and moves them to a new area, let's say to beginning of DNA molecule or to the end of a DNA molecule. And then duplication. Duplication repeats or duplicate one or few nucleotides within DNA molecule. And the last one is called base substitution. That means that certain nucleotide or few nucleotides will be removed and replaced with completely different nucleotide or nucleotides. Every time the chromosome is replicated, mistakes can occur, causing mutations. About one cell in hundreds million carries a mutation in any particular gene. This is what we call spontaneous mutations, and they are rare. Mutations can cause death of the cell, but some mutations can be very beneficial. For example, mutation might allow a cell to grow faster. As a result, mutated cells will soon dominate in population. And then induced mutations are more frequent. For example, in biotechnology labs, the frequency of mutations increased when culture is treated with a mutagen. There are different types of mutagens that can be used in biotechnology labs. For example, chemical mutagens, physical mutagens, or biological mutagens. An example of a physical mutagen will be UV light or sunlight and radiation. On this slide, they show you a picture of chemical mutagen, which is bromouracil. BU stands for bromouracil. Bromouracil has affinity to AT base on DNA molecule, and because it has affinity to AT base, it can remove AT base from certain region of DNA molecule and replace it with GC base. So that means bromouracil has ability to cause base substitution type of mutation. And biological mutagens, this is what we call transposable elements. They also have ability to cause different types of mutations. But let's go back to UV light or sunlight, which is an example of a physical mutagen. How does it cause mutations? When we are exposed to the sunlight too much, sunlight can cause damage by affecting our skin. DNA molecule in our skin, after it was treated by UV light, starts forming what we call dimers, thymine dimers. Dimer means two thymines form bond to each other. That will activate repair system of our immune system, and the immune system will come over and will remove those dimers and replace them with the normal thymines. But sometimes, for some reason, immune system is not able to fix that damage, and that results in mutation. As a result, our patient in this case will develop symptoms of skin cancer. Let's talk about consequences of mutations. Actually, two factors determine how serious a mutation will be for a cell. Number one, how much damage the mutation does to gene, and number two, how important the gene product is to actual cell. For example, missense mutation. When a missense mutation occurs, it means one amino acid in the protein will be replaced with different amino acid. If that new amino acid in the structure very similar to amino acid that's supposed to be in that region of DNA molecule, it results in change of function of protein, but that protein still will be able to do its job, just with few changes. But if cell develop nonsense mutation, in this case, amino acids will be replaced, let's say, with stop or nonsense codon. And because stop or nonsense codon doesn't encode any amino acid, that means basically that protein will have a hole in that area. And of course that will destroy protein completely, and that protein won't be able to do its job, because nonsense mutation completely destroyed activity of that particular protein. And on the final part of this lecture, we will talk about a genetic exchange. What is this genetic exchange? Genetic exchange is the transfer of genes from one cell to another. For example, for eukaryotic cells, a sexual reproduction will be an example of genetic exchange, when two cells share information with each other and form completely new cell. So we can say that it is actually essential process for eukaryotic cells. Bacteria, on the other hand, have ability to transfer or share only a portion of DNA molecule, and this is not essential for them. We start with the first type of genetic exchange. It is called transformation. It can be natural or artificial. Let's start with the natural transformation. Some bacteria have ability to pick up fragments of DNA molecule from extracellular space. Each piece of DNA carries a certain information. Bacterial cells can read that information and use it for their own benefits. Let's say, using this information from that particular fragment of DNA, this bacterium becomes resistant to one specific antibiotic. But what is going to happen if this bacterium divides and produces two new cells? If this fragment of DNA molecule is not incorporated into cell's DNA, as soon as it divides, it produces new cells. That piece of DNA molecule will be lost, and two new cells will not be able to use this information from DNA fragment. But if that piece or fragment of DNA molecule get incorporated into cell's chromosome, then when it divides and produces two new cells, both new cells will inherit this information and will be able to use this information for their benefits. Means they both will become resistant to that specific antibiotic. So that was an example of natural transformation. Now let's talk about artificial transformation. Artificial transformation is induced in our biotechnology labs. For example, you probably know that bacteria make insulin today for us. So how do they make that insulin? Why do they make that insulin? How do we make them to start making insulin for us? Basically what we do, we take a fragment of DNA molecule which encodes information how to make insulin, and we incorporate or insert that piece of DNA into chromosome of certain bacteria. Because that piece of DNA is incorporated into cell's chromosome, not only this cell is going to use that information and will start producing insulin, but after it divides and produces two new cells, both new cells also will have that fragment of DNA in their structure and will continue making insulin for us. The second form of genetic exchange in bacteria is conjugation. Do you remember on the first lecture I told you that bacteria have what we call sex pili or sex pilus, and I told you back then that bacteria use sex pili for communication. For example, today when our patients have a GI tract infection caused by bacteria, and if this infection is not life-threatening and patient is not immunocompromised, we prefer do not use antibacterial treatment to cure that infection. Why? Because most of the times those infections are not life-threatening and patients are able to recover by themselves. And the second reason why, because we have huge number of bacteria living in our GI tract, and they have ability to communicate. They use sex pili and share information. So if we use antibiotic to treat bacterial gastrointestinal infection, as soon as one bacterium within gastrointestinal tract of that patient becomes resistant to that antibiotic, they perform conjugation. As a result, all of them in short period of time become resistant to that antibiotic. As a result, patient becomes a carrier of infection. So let's get back to conjugation. This picture shows you how conjugation occurs. On this picture you see E. coli cells. This is E. coli F plus cell, means it has that plasmid, conjugative plasmid, and that means F plus cell is already resistant to antibiotic. This is E. coli F minus cell. You see there is no plasmid within cytoplasm. That means this cell is still sensitive to antibiotic. What they're going to do next is they're going to use these 6 spirals to attach to each other, and then F plus E. coli cell will give piece of plasmid to F minus cell. As a result, both cells become F plus cells, means both of them are resistant to antibiotic. And finally, the last form of genetic exchange in bacteria is transduction. They show you transduction on this slide based on the example how certain viruses replicate. They explain the replication of bacteriophages. Bacteriophage, it is a virus that infects bacteria. Means bacteriophages or phages, those are viruses that use bacterial cells as a host cell. Let's look at this picture. Here's some kind of bacterial cell. Here's our virus, bacteriophage. The first step, it will attach to the surface of host cell. Now it's already attached. The next step is going to insert its DNA molecule into host cell chromosome. Into host cell cytoplasm. So you see this is a viral DNA. It's already incorporated into cytoplasm of this particular host cell. What is going to happen next? Next, that piece of viral DNA will be incorporated or inserted into a chromosome of this host cell. As soon as it happens, this cell will be turned into what we call provirus. It means that bacterial cell will start making parts of that particular virus. Then those parts will be assembled into mature viruses, and when they're mature enough to leave host cell, they destroy it, and this way they get released. If you look at this picture, all those new viruses that are made by this host cell have DNA of initial bacteriophage or virus, except this particular virus here on the right side. For some reason, this one virus inherited DNA of the host cell. So that means next step, when that virus will try to replicate, it will find host cell again. You see it is bacterial cell again. It will attach to the surface. It will insert its chromosome or DNA into cytoplasm of the host cell. Then it will be incorporated into host cell chromosome. But what is going to happen next? Nothing. Because that piece of DNA is actually bacterial DNA, this host cell will accept that piece of DNA and will use that information on DNA, piece of that fragment of DNA for own benefits, but that pretty much the end of the road for this particular virus. So this is what we call transduction type of genetic exchange.